Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Seed germination and dormancy, from soil to sprout. Table of contents. Introduction to seed germination. Why is seed germination important? Factors affecting seed germination. Common seed dormancy mechanisms. How to break seed dormancy. Methods to test seed viability. Tips for successful seed germination. Common issues in seed germination. Conclusion. Introduction to seed germination. Seed germination is the process in which a seed begins to sprout and develops into a new plant when the environment is appropriate. It marks the stage where a dormant seed transforms into an active seedling that will grow into a fully developed plant. The process starts when the seed absorbs water, imbibition, causing it to swell. Water absorption activates enzymes that break down stored nutrients within the seed. The breakdown of these nutrients provides the energy needed for the radicle, initial root, to emerge. After the radicle appears, the plumule, shoot, begins to emerge. The seedling then establishes itself by forming a root network and starting the process of photosynthesis. Why is seed germination important? Seed germination is essential for plant growth, farming, and maintaining ecological balance. It ensures plant reproduction and continuity by allowing species to persist and adapt to changing environmental conditions. Successful germination is critical for food production and agriculture, as crops like wheat, rice, and maize depend on it for higher yields and food stability. It contributes to forest regeneration and biodiversity by enabling natural reforestation and supporting wildlife ecosystems through diverse plant growth. Germinated seeds help in soil conservation by stabilizing soil particles and preventing erosion, while also regulating the climate by absorbing carbon dioxide. Many germinated plants, such as neem, tulsi, and aloe vera, hold medicinal value, and economically important crops like coffee, cotton, and tea depend on proper seed germination. It plays a significant role in scientific research and biotechnology, helping to enhance crop resilience, boost food production, and develop drought-tolerant plant varieties. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Phases of seed germination. Imbibition, water absorption. The first stage of seed germination where the dry seed absorbs water, causing it to swell and initiate metabolic activity. The outer seed coat softens, allowing water to reach internal tissues and activate enzymes. Water uptake provides necessary moisture for biochemical processes that support growth. Increased seed volume builds internal pressure, eventually rupturing the seed coat and enabling embryo development. Enzyme activation and metabolism. After water absorption, enzymes are activated and metabolic rates increase to support germination. Stored nutrients like starch, proteins, and fats in the endosperm or cotyledons are broken down by enzymes such as amylase, protease, and lipase. These simpler nutrients supply energy for embryo cell expansion and division. Aerobic respiration begins, producing ATP needed for the growth and development of the seedling. Radicle emergence, primary root growth. The radicle, or primary root, is the first visible sign of germination as it breaks through the seed coat. It anchors the seed into the soil and absorbs water and nutrients, forming a strong foundation for the plant. Root hairs develop to enhance the plant's absorption capacity and support further growth. Plumule emergence, shoot growth. Following radical growth, the plumule or embryonic shoot begins developing upward toward the surface. In hypogeal germination, cotyledons stay underground. In epigeal germination, they emerge above the soil. The plumule eventually forms the stem and leaves, initiating photosynthesis. A protective covering, coleoptile in monocots or hypocotyl in dicots, shields the plumule during emergence. Seedling establishment. The seedling transitions from dependence on stored food to independent nourishment via photosynthesis. True leaves appear and begin synthesizing carbohydrates using sunlight, water, and CO2. The root system expands, enhancing nutrient and water uptake and supporting growth into a mature plant. Factors affecting seed germination. Water. Water is essential for seed germination as it triggers imbibition, activates enzymes, and stimulates metabolic activities. Seeds cannot germinate without sufficient moisture, while excessive water may cause fungal growth, seed decay, or oxygen deficiency. Different seeds have varying water needs, so maintaining the right soil moisture level is key for successful germination. Oxygen. Oxygen is necessary for aerobic respiration, which generates the energy required for seed development. During germination, seeds shift from a low to a high metabolic state, increasing their need for oxygen. Poor aeration, deep sowing, or waterlogged soil can result in oxygen deficiency and hinder germination. Well-aerated soil ensures adequate oxygen supply for growing seedlings. Temperature. Temperature influences the rate of enzyme activity and metabolic processes, with each plant species having an optimal temperature range. Low temperatures slow down enzyme activity, while extremely high temperatures can denature enzymes and inhibit seed growth. Some seeds, such as those of winter crops, require vernalization, exposure to cold, to germinate, while tropical seeds prefer warmer conditions. Light or darkness. Certain seeds have specific light requirements to sprout, with photoblastic seeds like lettuce and tobacco needing light, while others like onions and violets germinate in darkness. Light affects the production of growth hormones such as phytochromes, which play a role in breaking seed dormancy and initiating germination. Seed dormancy. Seed dormancy is a natural mechanism that prevents premature germination in unfavorable conditions. Dormancy may be due to a hard seed coat, presence of biochemical inhibitors, or an underdeveloped embryo. Overcoming dormancy through various methods ensures proper germination when environmental conditions are suitable. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Common seed dormancy mechanisms. Physical dormancy, hard seed coat. Certain seeds, such as those from lotus plants and legumes, possess a water-resistant seed coat that restricts the entry of water and oxygen. This tough seed coat serves as a shield against unfavorable conditions, preserving the seed until suitable circumstances arise. Mechanical scarification, a process that involves scratching, cutting, or softening the seed coat, breaks physical dormancy and allows water to enter. Physiological dormancy, chemical inhibitors. Some seeds have chemical substances, like abscisic acid, which postpone germination until they are broken down or washed away by water. These substances impede early sprouting and ensure germination occurs at the right moment. For example, tomato and citrus seeds require adequate moisture to eliminate inhibiting chemicals before germination. Morphological dormancy, immature embryo. Seeds are released from the parent plant while still containing an undeveloped embryo in certain species. These seeds require additional time to complete their growth before they can sprout. Plants that exhibit this type of dormancy include ginkgo and orchids, where further embryonic growth is stimulated by warm and moist environments. Combination and dormancy, multiple mechanisms. Certain seeds exhibit both physical and physiological dormancy, making them particularly challenging to germinate. For example, wild cherry seeds possess a tough seed coat, accompanied by chemical inhibitors, and require several treatments to break dormancy. These seeds can undergo both scarification and stratification to promote germination. How to break seed dormancy. Scarification, breaking the seed coat. Scarification involves weakening or breaking the hard outer seed coat to allow water and oxygen penetration. It can be done mechanically, e.g., sanding or nicking the seed, chemically, e.g., soaking in acids, or thermally, e.g., dipping in hot water. This method is commonly applied to seeds with hard coats like peas, acacia, and morning glory. Stratification, chilly or mild treatment. Some seeds require temperature treatments to mimic seasonal changes and overcome dormancy. Cold stratification involves storing seeds in cold, moist conditions, 1 to 5 degrees Celsius, for several weeks or months, as required by seeds like apple and maple. Warm stratification is used for tropical seeds like cocoa, where seeds are kept in warm, humid environments before planting. Water soaking. Soaking seeds in warm water for a few hours or overnight is a simple and effective method to soften seed coats. This process also helps leach out chemical inhibitors that might prevent germination, commonly used for seeds such as beans, lentils, and coriander. Light or dark exposure. Some seeds are photoblastic and require specific light conditions to germinate. Light-sensitive seeds like lettuce and tobacco need exposure to light to sprout. Others, like onions, require complete darkness to initiate germination. Adjusting light exposure based on seed requirements enhances germination success. Hormone treatment. Plant hormones such as gibberellic acid are used to stimulate germination by overcoming physiological dormancy. Gibberellic acid promotes faster and more uniform growth by breaking chemical dormancy barriers. This approach is especially useful in horticulture and agriculture to ensure consistent seedling production. Methods to test seed viability. Seed viability refers to a seed's ability to germinate and grow into a healthy plant under suitable conditions. Testing seed viability is important in agriculture, horticulture, and research to ensure successful crop or plant development. A range of methods is available, from simple home-based tests to advanced laboratory techniques. The water float test. One of the simplest ways to test seed viability. Seeds are placed in water and left to soak for a few hours. Viable seeds are denser and typically sink, while non-viable seeds float due to internal air pockets. This test offers quick results but is not fully reliable, as some viable seeds may still float. Germination test. The most common and user-friendly method for determining seed viability percentage. A known quantity of seeds, e.g., 50 or 100, is placed on a moist paper towel or cotton pad. Seeds are kept in a warm place for several days. The number of seeds that successfully sprout reflects the viability rate, e.g., 80 sprouts out of 100 seeds equals 80% viability. Tetrazolium test, TZ test. A biochemical test that detects seed viability through enzymatic activity. Seeds are soaked in a colorless tetrazolium chloride solution. Living cells react and turn red, showing active tissue. The intensity and pattern of red coloration indicate the seed's health and potential. Commonly used in seed laboratories due to its speed and reliability. Cutting test. A simple but destructive method for checking seed health. Seeds are cut open to inspect their internal structure. Healthy seeds appear white, firm, and well-developed. Non-viable seeds may be empty, shriveled, or discolored. Since the seed is damaged during the test, it cannot be used for planting afterward. X-ray test. A high-tech, non-destructive method used mainly in research and seed production facilities. Seeds are exposed to X-rays to visualize their internal features. Helps identify hollow seeds, insect damage, or developmental defects. While highly informative and non-invasive, it requires expensive equipment and technical expertise. Tips for successful seed germination. Achieving successful seed germination requires careful attention to environmental conditions, seed quality, and proper handling techniques. By applying the following best practices, you can significantly improve germination rates and support healthy seedling growth. Use fresh and viable seeds. Seed viability declines with age. Always verify expiration dates on seed packets and store them in a cool, dry location to preserve their ability to germinate. If you're using stored seeds, consider conducting viability tests beforehand to ensure successful sprouting. Maintain proper moisture levels. Seeds require steady moisture to initiate germination, but overwatering can lead to fungal infections or rot, while too little water may halt the process. Light misting with a spray bottle helps keep the soil evenly moist without causing saturation. Ensure optimal temperature. Each type of seed has a specific temperature range for ideal germination. Warm season varieties like peppers and tomatoes sprout best between 20 to 30 degrees Celsius, while cool season crops like lettuce prefer 10 to 20 degrees Celsius. Use a heat mat or greenhouse to maintain suitable temperatures when needed. Promote good soil. Aeration. Oxygen is vital for seed respiration. Compacted soil can block airflow and restrict root development. Choose light, well-aerated soil or seed starting mixes to enhance both aeration and moisture retention. Protect seeds from diseases and pests. Seed-borne pathogens, fungi, and mold can negatively impact germination. Use sterile soil, ensure good air circulation, and apply appropriate fungicides if needed. Some seeds may benefit from soaking in hydrogen peroxide or potassium permanganate solutions before planting. Sow at the correct depth, Planting depth should match seed size. Larger seeds, such as beans or corn, should be buried 2 to 3 centimeters deep, while smaller seeds like lettuce and basil should be lightly covered or left on the surface, especially if they need light to germinate. Provide adequate light. Some seeds, known as photoblastic, need light to germinate, while others do better in the dark. Knowing the specific light requirements of each plant variety is key to improving germination rates. For indoor setups, supplemental grow lights can boost natural lighting conditions. Common issues in seed germination. Several factors can hinder successful seed sprouting, but identifying these problems early can significantly enhance germination success and seedling development. Failure to germinate. Seeds that do not sprout may be old, non-viable, planted at incorrect depths, or exposed to unsuitable moisture and temperature levels. To resolve this, test seed viability beforehand and ensure optimal planting conditions. Damping off disease. This fungal infection causes seedlings to rot at the base and collapse. It is commonly triggered by excessive moisture, poor air circulation, and contaminated soil. Prevent it by using sterilized seed starting mix, avoiding overwatering, and ensuring good ventilation. Uneven germination. When some seeds sprout while others don't, the issue may stem from inconsistent watering, temperature fluctuations, or uneven seed quality. Use high-quality, uniform seed lots and maintain steady environmental conditions to ensure even germination. Leggy seedlings. Seedlings that grow tall and spindly often suffer from insufficient light. This is especially common indoors, when natural sunlight is limited. Place seedlings closer to a light source or use reflective surfaces to improve light exposure and encourage sturdy growth. Mold on soil surface. White or fuzzy mold on the top soil indicates high humidity and poor airflow. Combat mold by dusting the soil with cinnamon, a natural antifungal, reducing watering frequency and enhancing ventilation. Poor seedling development. Post-germination. When seeds sprout but fail to thrive, it may be due to nutrient deficiency, irregular watering, or poor soil conditions. Transplant seedlings into nutrient-rich soil and apply a balanced fertilizer to support vigorous growth. Conclusion. Seed germination marks the beginning of a plant's life cycle, involving stages such as water uptake, metabolic activation, emergence of the radicle and plumule, and seedling formation. The success of germination is influenced by factors like water availability, oxygen, temperature, light, and the absence of dormancy barriers. Some seeds germinate naturally under favorable conditions, while others require dormancy-breaking methods, such as scarification, stratification, or hormonal treatments. Testing seed viability is essential in agriculture and horticulture to predict germination potential using techniques like the float test, germination test, cutting test, tetrazolium test, and X-ray analysis. Ensuring proper environmental conditions, selecting high-quality seeds, and addressing common issues like damping off, mold, or leggy seedlings significantly enhance germination success. A clear understanding of germination processes and challenges enables better decision-making for farmers, botanists, and gardeners, leading to healthy plant growth, improved yields, and effective reproduction. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!